Welcome back to the next video in our introductory series looking at the immune response. In this video, we're going to continue our preliminary discussion of adaptive immunity in particular, talking about a specialized process called antigen presentation. This video is going to be a very broad and sort of simplified overview of this process, and we'll dig into it a lot deeper as we get further into the course. But today's going to help us learn some of the introductory terminology and just get a big picture of this process, because it's going to help us understand some of the specialized functions and needs that T cells have in particular in order to mount an adaptive immune response. So let's dig into antigen presentation. Antigen presentation is specialized, or it's necessary, I should say, because T cells have this specialized requirement or challenge, if you will, that they are unable really to recognize antigens on their own. So we talked about how cells need to recognize self versus non-self, and in particular, adaptive immune cells need to recognize a particular non-self signal called antigen. T cells recognize antigen. That's very key to their full activation. However, they're not really able to interact with an antigen directly, physically sort of go up to a bacterium or a virus and see it for what it is and know that they're supposed to respond to it. They require some help from some other cell types, as we'll see, and that process of getting help from other cell types is what is known as antigen presentation. And this is a way, in fact, that we bridge innate and adaptive immunity, because the cells that tend to present antigen to T cells are generally cells of the innate immune system. These innate immune cells, or any cell that is presenting antigen, we can refer to as an antigen-presenting cell, or APC. That'll be a very important abbreviation going forward in the course. And as you can see here in this cartoon, APCs basically instruct T cells when an antigen is present. They are sort of molecular translators or teachers, if you will. And T cells, when they are instructed that an antigen is present through the activity of an antigen-presenting cell, can then become fully activated and mount what we would call their effector functions, the type of biological work they can perform that is somehow antimicrobial or somehow otherwise immunomodulatory. So what do antigen-presenting cells do in the process of antigen presentation? Well, there are many ways that antigen presentation can occur. A major one that perhaps is most famous is through the presentation of antigens that APCs have taken in through the process of phagocytosis. And this is sort of a classic example of how we bridge innate immunity into adaptive immunity, because, as we said in the last lecture, many innate immune cells tend to be phagocytes. They are cells that are good at eating pathogens and damaged cells, other types of entities in their environment that we need to mount some sort of immune response against. So in this particular example of a way that innate immune cells can facilitate antigen presentation, an APC is going to detect a pathogen through pattern recognition receptors. And remember that innate immune recognition of non-self tends to happen through pattern recognition rather than through antigen recognition. So right now we're at the very beginning. The antigen-presenting cell sees some sort of pathogen and uses a pattern recognition receptor to detect a pathogen-associated molecular pattern, or PAMP, on the surface of that pathogen in order to know that there is a non-self entity in its presence. And often when that occurs, that innate immune cell is going to engulf and eat that pathogen through a process that we call phagocytosis, which maybe you've seen in the context of other courses. Basically take it in to an intracellular vacuole system of some kind, and that vacuole is going to merge with the lysosome, and ultimately what's going to happen there is that through the process of acidification and the activity of protease enzymes, it's going to break down that pathogen and take the proteins and other macromolecules associated with that pathogen and sort of chop them up, if you will, into small fragments. And the small fragments of proteins will be especially important because small, short amino acid sequences or peptides are what are actually going to serve as antigens for the purposes of antigen presentation. So you can see here in a structure that we call the phagolysosome, inside of this antigen-presenting cell, you have some broken down pathogen bits, some of which are short polypeptide sequences which can serve as antigens. And the antigen-presenting cell is then going to take those peptides peptide sequences back out of the phagolysosome system and traffic them back to its own cell surface. And as you can see here again, simplified in cartoon form, sort of put it on its surface in order to show it to a T cell in its environment. And when a T cell sees an antigen not directly associated with the pathogen, but instead when it sees that antigen associated with an antigen-presenting cell, it is then able to recognize it, become activated, and mount its effector response. And so, why does it matter? Why do T cells, why can they only see the antigen on the APC and not on the pathogen itself? Well, a major reason for that is that T cells do not recognize antigen alone, but they recognize antigen in combination with another important immunological molecule called MHC. MHC is short for Major Histocompatibility Complex. We'll learn about the origin of that name later in the course, but for now, just think of these molecules as antigen-presenting molecules. And there are two classes of them, as we say, MHC class 1 and MHC class 2. And as you can see here, MHC class 1 and MHC class 2 are very similar molecules. They have a similar structure, although you can see some structural differences, which again, we'll get into a bit later. But what they share in common is what we call a peptide binding groove or cleft. Again, a complementary shape that is able to hold an antigen and sort of stick it out into space where it is available then to be interacted with by a T cell in the environment. And so MHC class 1 and MHC class 2 tend to present different types of antigens, and as we'll see, they tend to activate different types of T cells. But the purpose of this slide is just to say that T cells require antigen to be associated with MHC in order to see it, in order to interact with it. They cannot bind to an antigen that is just sort of floating around as a soluble molecule in the extracellular environment, or even sort of stuck to the surface of a pathogen, because MHC is a host protein. We only find it on our own cells. MHC is not found on the surface of pathogens. So what cells express MHC, and how does that influence the way that different types of cells can activate different types of T cells? Well, something very important to know is that all nucleated cells, in fact, express MHC class 1, which is shown here as this molecule designated in purple. And what I'm getting across with these different cell types is that, you can see here an epithelial cell, a neuron, a muscle cell, they all express MHC class 1. This is extremely widely expressed throughout the body, and for that reason, almost any cell can be an antigen-presenting cell. Almost any cell has MHC class 1, and in fact, in humans, all nucleated cells basically means every cell except for red blood cells, which are anucleated in humans and most other mammals. So antigen-presenting cells don't necessarily have to be immune cells. They can be cells all over the body. And as we'll see, that's important because cells all over the body can be infected. And in fact, that is really what MHC class 1 is for. When a cell becomes actively infected, when it has some sort of pathogen, especially something like a virus, which is an intracellular pathogen, it's replicating inside the cell, that cell is able to take proteins associated with that pathogen, load them onto MHC class 1, where then a T cell is going to be able to recognize the antigen through its own antigen receptor, which we call the T cell receptor, or TCR, another very important abbreviation. So the TCR is the receptor on the surface of a T cell, which is its means of detecting non-self, particularly antigens. So as you can see here, there's an interaction between all three of these components, MHC class 1, the antigen, and the TCR. We need all of these components to sort of put together those complementary shapes in order to have the full sort of interlocking interaction and to transduce a signal into the T cell and activate it. So again, a major thing that I want to highlight here is that because MHC class 1 is expressed on virtually all cell types, this is the main way that we detect actively infected cells anywhere in the body. However, there are other types of antigen presentation. We just said that there's another type of MHC, so let's talk about it. A key concept in antigen presentation is that there are actually specialized antigen presenting cells, or APCs, that we call professional antigen presenting cells. Here, sort of designated by this dendritic cell, a type of immune cell, sort of wearing a professional outfit. And as you can see on its surface, it is expressing both MHC class 1 and MHC class 2. So professional APCs uniquely express MHC class 2. So only professional APCs express MHC class 2, but they all also express MHC class 1. So there's not any cell type that only expresses MHC class 2 under normal conditions. They either only have one or they have both 1 and 2. And the ones that have both we refer to as professional antigen presenting cells. And generally, the types of antigens that are presented on MHC class 2 are not from an active infection inside the cell, but instead are coming from those phagocytosis-derived antigens that we mentioned a few slides back. So professional APCs tend to be very good phagocytes. They're very good at eating pathogens, digesting them in their phagolysosomes, and then trafficking those antigens and presenting them via MHC class 2 on their cell surface, even if the cell itself is not actively infected. So what does it matter whether or not an antigen is presented on MHC class 1 or MHC class 2? The key outcome, or the key difference in the way this facilitates a T cell mediated immune response, is that there are unique populations of T cells that recognize antigens only through interactions with MHC class 1 or only through interactions with MHC class 2. So these two major types of T cells that I just mentioned, you can see designated here. We have cytotoxic T cells, which are sometimes designated as T subscript C or TC cells. And as their name implies, they are cytotoxic, meaning they can kill a cell, cyto meaning cell. And you can get the idea here with this sort of angry-looking T cell with a sword. He's ready to kill something. In contrast, we have other populations of T cells called helper T cells, or T subscript H. And T helper cells, they are not directly cytotoxic. Instead, they modulate the immune response through the secretion of soluble signaling molecules, and we'll say more about that in just a second. For this slide, though, what I want you to key in on is that cytotoxic T cells interact with antigen, recognize antigen, only when it is being presented on MHC class 1. So cytotoxic T cells are able to recognize antigen being presented by almost any cell, because almost any cell expresses MHC class 1, as we've said. So they're very good at detecting actively infected cells, even non-immune cell types, but they can also be activated by professional APCs as well. However, helper T cells only recognize antigen being presented by MHC class 2, and for that reason, the only way that we can activate a helper T cell is through presentation by a professional APC, which again uniquely express MHC class 2. So, and that's designated with these additional text boxes here. So I'll repeat it, is that most cells, actually this is sort of a mirror image of what I said, most cells can only activate cytotoxic T cells when they're infected. In contrast, professional APCs can activate both cytotoxic T cells as well as T helper cells. So why is it that the cytotoxic T cell can only recognize antigen on MHC class 1 and a T helper cell can only recognize antigen on MHC class 2? The molecular reason for this is that both of these populations of T cells each express their own unique T cell co-receptor. So a cytotoxic T cell expresses, in addition to its TCR, its T cell receptor, which recognizes antigen, a co-receptor called CD8, or cluster of differentiation 8. And CD8 is actually an equal part of the overall molecular shape that I mentioned that allows all of these molecules to interact in the presence of antigen. So a cytotoxic T cell, because it expresses CD8. CD8 is complementary to MHC class 1, and that's the molecular determinant, basically, that sort of confers the specificity for MHC class 1 onto cytotoxic T cells, because they only have CD8, they can only interact with MHC class 1 in order to facilitate the recognition of antigen. In contrast, T helper cells express a co-receptor called CD4, and by a similar logic, CD4 has a high degree of specificity for MHC class II. And again, we sort of have this multipartite molecular interaction involving all of these molecules. And in the absence of CD4, the TCR is not able to interact with MHC class II. But when CD4 is present, you get a full interaction, and again, CD4 confers specificity for MHC class II, and therefore T helper cells can only recognize antigen from cells that express MHC class II, which, of course, as we've said, are professional APCs. So the expression of these co-receptors, CD8 and CD4, are sort of the definitional molecules on the surface of these cells that really help us identify whether or not a T cell is a cytotoxic T cell or whether or not it's a T helper cell. And in fully mature adult T cells, they only express one or the other. And we'll say, again, another thing that we'll come back to a little bit later in the course. But for the purposes of our first week, cells either express only CD8 and therefore are only cytotoxic, T cells, I mean, or they only express CD4 and are therefore T helper cells. So what is the difference then in effector function between a cytotoxic T cell, or in fact, I could say sometimes we call cytotoxic T cells CD8 T cells, or sometimes even just CD8s. In fact, that's extremely common. Knowing which type of cell expresses which co-receptor is very important for that reason, so you can sort of keep up with the lingo of the way that we refer to T cells, and I'm sure I'll do that in these lectures. In contrast, T helper cells are frequently referred to as CD4 T cells or just CD4s. So using that then, what is the difference between the effector function or the biological functions performed by CD8 T cells or CD4 T cells? Well, as we just discussed, cytotoxic T cells are cytotoxic. Their main effector function is actually to induce cell death within actively infected cells. And so as you can see in this cartoon, you have some kind of surface of cells, and the cell in the middle is infected with something here. We can imagine maybe it's some kind of intracellular bacterium. That infected cell is going to take peptides, take antigens associated with that bacterium, and present them via MHC class I, which we can see here in purple. When they do so, that is going to facilitate the activation of a cytotoxic T cell, a T cell expressing the co-receptor CD8. And using both its TCR and the co-receptor CD8, it's going to be able to dock onto that MHC class I molecule, and the antigen is going to be able to activate the TCR, which is going to transduce a signal into the cytotoxic T cell and activate it. And when a cytotoxic T cell becomes activated, it releases molecules, soluble molecules, as well as other types of molecules on its surface as it's physically interacting with the infected cells, which in some are going to induce apoptosis or programmed cell death in the target cell. So the CD8 T cell induces the process of cell death, which you can see playing out here. And this is important because generally cell death is a pretty good strategy for stopping an infection in its tracks. Infections tend to, when they occur, especially an intracellular infection, that pathogen is using the resources of the infected cell. It's using its metabolism. It's using its ribosomes to make its proteins so that it can replicate itself. And so the infected cell is really sort of a factory that is producing more of that bacterium or virus or whatever it is. And so by killing the cell, we can basically remove that cell as a replicative niche for the pathogen. And this is usually a pretty good strategy. So it's very important generally to kill cells when they are infected. And most cells are easily replaced. They can undergo mitosis, the healthy ones can, and you can sort of replace them over time with healthy uninfected cells. And importantly, T cells only kill cells which are expressing the antigen that they recognize. And so you can see here that the healthy cells nearby that are uninfected and therefore not presenting that pathogen-derived antigen, they are spared from the cytotoxic effects of the T cell. And so this is a key idea in immunology, is that the process of antigen presentation is helpful because it makes sure that T cells are only inducing, or cytotoxic T cells in particular, only inducing cell death exactly where it needs to happen and not harming healthy bystander cells. which might occur if we had a less regulated or less complicated process for activating the CD8 T cell. So enough for now to say that cytotoxic T cells, their main effector function is the induction of programmed cell death or apoptosis in actively infected cells presenting antigen via MHC class I. In contrast, T helper cells, which again express CD4, they do not directly induce death in infected cells. And remember that they are recognizing their antigens through through professional antigen presenting cells, which may or may not be actively infected. In fact, most of the antigen derived or being presented by professional APCs actually came in through phagocytosis rather than direct intracellular infection. And so when the professional APC presents its antigen to the CD4 T cell and activates it through its TCR, the T helper cell is generally going to sort of help other immune cells in its environment, which is where its name comes from. And the way that it does this, oversimplifying a bit, is through the secretion of molecules that we call cytokines. Cytokines are soluble chemical messengers of the immune system, which allow immune cells to talk to each other, basically, and sort of modulate each other's activity in ways that usually facilitate some sort of antimicrobial function. So a few examples that you can see here on this slide is that when the T helper cell becomes activated, it starts secreting cytokines, and they might do any number of things to other immune cells in the environment. So for example here, this looks like a cell called a macrophage, which is a very good phagocyte. And these cytokines might turn up the process of phagocytosis in that macrophage, allow it to sort of eat these bacteria even more effectively, process them for antigens, which is then going to facilitate further antigen presentation downstream. So that could be very helpful. T helper cell derived cytokines can also induce processes of proliferation and differentiation in other types of immune cells, sort of determining how many and what types of immune cells we have available in any given immune response in order to fight an infection. They can also induce the effector functions of other immune cells, which we'll be introduced to in the next lecture. And so this is an example of a cell called a neutrophil, and cytokines derived from T helper cells, for example, might induce the neutrophil to release its own cytotoxic molecules that it actually stores within its own cytoplasm. And so instead of the T helper cell directly inducing cell death, it can activate other cell types which are capable of cytotoxicity, and therefore indirectly sort of fight the pathogen, again, through helping facilitation of the effector functions of nearby immune cells. And again, as I've said many times, much more about all of these processes in future lectures. But for now, let's summarize this bird's eye view that we just went through for antigen presentation. I wanted to stress that antigen presentation bridges the innate and the adaptive immune systems because, to a high degree, antigen presentation occurs through the activation of an innate immune cell through a pattern recognition receptor, which is going to induce it to undergo phagocytosis, or maybe it's just going to induce it to present antigen via MHC class I if it's actively infected. But nevertheless, that process of antigen presentation is then going to activate T cells in the environment, which is going to be key for an effective adaptive immune response. We learned that antigens are presented via molecules called MHC, and that there are two classes of MHC, MHC class I and II, and we saw that MHC class I is expressed by all nucleated cells in the body, whereas MHC class II is uniquely expressed on professional antigen-presenting cells. And this mattered because this determined the types of T cells that were going to be activated when antigen presentation occurs. So cytotoxic T cells, which express CD8, are activated by antigen being presented on MHC class I, whereas T helper cells, which express CD4, can only be activated by antigen expressed on MHC class II. Finally, the type of T cell that is activated by antigen is going to determine the effector function that occurs. So CD8 T cells have intrinsic cytotoxic activity. They can directly kill infected cells in their environment. Whereas CD4 T cells, or helper cells, tend to modulate the activity of other immune cells in their environment through the secretion of soluble mediators called cytokines. So in our next video, we will continue our introductory discussion by learning about many other types of immune cells, some of the ones that we were just introduced to, some of the other cell types, for example, that CD4 T cells are able to help, and get a much more comprehensive pictures of all of the cellular players in the immune response. See you then.